Yes.
So, thank you very much,
Yes.
Danuta, for this introduction. Well, to start, I'm not really a specialist of wine. I mean, I work in a lab where we work with wine, but I'm not the specialist of wine. So, I am from the physical chemistry of food and wine team. It belongs to the PAM laboratory, which itself is attached to two entities, Agrosupdijon. It's an engineering school that forms agronomists or there is something that, okay, or agro-food engineers, and the University of Bourgogne-Franche-Comté. So in the world, wine occupies a preponderant place in various cultures. It participated and still participates in the rituals of various religions. It is appreciated by a large part of the populations. It is considered as a real social bond, and it has been so since ancient times. From the very beginning, early humans picked berries. And if they were stored in any wooden or stone container, after a few days with fermentation setting in, juice at the bottom of the container would naturally begin producing a low alcohol wine. According to recent archaeological, chemical, and botanical data, winemaking emerged in the Neolithic, with the transition from a nomadic to a sedentine style of living, which led to agriculture and vine domestication. The earliest data are now from the Near East, where the wine was in all probability used for the first time and gradually spread to Egypt and Mesopotamia. Then to Crete and to the coast of Europe and the area of Caucasus. This slide shows a nice fragment of a fresco where the harvest and the winemaking is represented. These Egyptians squeezed the grapes, collecting the flowing juice and save it in amphorae for maturation and conservation. All this industry was found in the oldest known winery discovered in the Arena cave of the village of Areni in Armenia. This site contained a wine press where the grapes were pressed on this tilted bowl. The juice can flow in a buried container where it can be collected and stored in fermentation vats and eventually in conservation jars. So this slide represents the wine production in 2018. Italy is #1, and it has been generally the case over years. Followed by France and Spain. Then we shift to the American continent, United States, Argentina, Chile. Then we go to Australia, China, Germany, South Africa, and we finished with Europe, Portugal, Romania, Russia, Hungary. Over the last decade, wine industry produced around 20 billion liters of wine per year. In 2018, on the world level, the associated trade was estimated to 30 billion euros. This activity and its direct or indirect repercussions in terms of work, regional planning, tourism, and so on, are, to say the least, significant. In practical, wine can be made from any form of fermented fruit, bananas, apples, cranberries, blackberries. But in most cases, wine is produced from grapes that are different than those usually found in supermarkets. Wine grapes varieties are smaller, sweeter, contain seeds, and have thicker skins. Each type of known wine, Cabernet, Merlot, Chardonnay, Pinot, comes from a single wine species called European grape wine. From a chemical point of view, wine is an ethanol solution that contains a wide range of components. We are talking here of tens of thousands of components, like organic acids, amino acids, sugar, aromatic molecules, minerals, and so on. In popular or advertising culture, many virtues are associated with moderate wine consumption. Here, an Australian company emphasizes 10 of these. I highlighted the one of reducing stress, something that one can feel after or during the consumption of a single glass of wine. So, if wine is such a relaxing agent, what else but relaxometry to check this point? Well, this is an easy and not very funny joke, but more seriously, the real reason why we did relaxometry is simply that we had the opportunity to participate to the cost action Eurolax. And I have to thank Danuta Crook, but I obviously want to thank all the people who worked for this action. And I also thank all the specialists of relaxometry who gave remarkable presentation during the meetings of the cost action or through these webinar sessions. As far as we are concerned, you must understand that before the cost action, relaxometry was an unknown area for us. And thanks to Eurolax, we have started to become aware of all that relaxometry has to offer in the field of foodstuff and packaging. To that respect, I want to say that for us, the cost action has been a great success. Our story began at the first meeting in Holstein. At the end of a food session, I went to see Adam Rashoki from the Molecular Dynamic Laboratory in Poznan, Poland, and just asked him, would it be possible to come to your lab and try to run some experiment on our packaging and foodstuff? Adam, who had never heard of me, just answered, yes, of course, you are welcome. So a few weeks later, barely not afraid of anything, I took different samples from my lab, biofilms, cork, but also cereals, proteins, and wine, and went to Poznan, where I was indeed very well received. I was able to perform many experiments on the sample brought and get informative data. And I will now present our results on wine. Here is a type of proton profile we obtained for wine. The sample is a red wine from Bordeaux, a blend of Medoc. We carried out a classical experiment, that is to say, we measured the longitudinal relaxation time as a function of the Larmor frequency of the proton. And we reported the relaxation rate. It's the inverse of the relaxation time. What happens at first glance, it's the occurrence of two features, two dispersions around 2 and 0.1 megahertz. Okay, before going any further, I just want to open here a short bracket for people or students, and I am especially thinking of those of my lab who are not familiar with NMRD profile. Let's take a simple water solution. When placed in a magnetic field, BO, and excited by some radio frequency impulsions, many interactions can take place. I shall only consider the dipolar interactions with it, which is a through space interaction between two protons separated by a length r. When the dipole is moving in the static magnetic field, it can relax. I mean, the dipole can release the energy stored during the application of some previous impulsions. Bloembergen, Purcell, and Pound have proposed a model for this relaxation. And after calculations, allow to express the relaxation rate in function of a dipolar constant that contain proton characteristics and the internuclear distance, the Larmor frequency nu of the proton, and tau correlation times that correspond to the time it takes to the dipole to rotate of one reagent as shown on this animation. The BBP model is composed of two terms. The first one in black, it's a pure Lorentzian line shape that is reported here. The blue one is not but nearly Lorentzian line shape, and the overall signal is the sum of the two previous contributions in green. In relaxometry, in practical, we don't consider the negative frequencies. So we keep only the positive half of the signal, of the curve. Moreover, as the frequency is very large, we use a logarithmic scale, and we end up with a profile that is now more similar to the observed profile on wine, with a plateau at low frequency and a dispersion at higher frequency. The red line and the red point correspond to the same frequency domain, but simply represented on a linear or logarithmic scale. For this profile, one can estimate the correlation time by refinement, or simply when the equation is simple like here, by measuring the frequency at half 8 of the signals and applying simple relation. I'm now closing the bracket and turn back to our wine profile. Starting from the beginning, when you just look at a glass or a bottle of wine, you realize that sometimes you may see some leaves, particles in suspension or crystals in the wine. Having in mind that big molecules like proteins moving in a solution because of the Brownian motion could give the type of signature observed on the wine profile, we wanted to check if these observed singularities are due to the dynamic of some macromolecules or some proton-containing particles present in the wine. If we solely consider a rotational movement, then in this case, a useful relation can be derived from the Stock-Einstein relation. It expresses the rotation time of the dynamic of the particle in function of the viscosity eta of the wine and the hydrodynamic radius of the particle. One can see that the larger the macromolecule or the viscosity is, the greater the correlation time and the slower the rotation is. Usually, at room temperature, the viscosity of wine varies between 2 and 6 millipascal per second. Assuming a rotational diffusion model, it is possible, as seen previously, to directly estimate the correlation times. And thanks to the Stock-Einstein relation, we can estimate the radius of the particle. 2 nanometers for the first dispersion and 5 nanometers for the second one. This dimension could correspond to a polysaccharide aggregate. The amount of polysaccharide in wine may be estimated between 200 and 400 milligrams per liter. Shape and size may vary a lot, but can reach up to 100 nanometer. 2 nanometer particle could also correspond to a protein of 30 kilodalton. In wine, we may find proteins from few kilodaltons up to 60, 80 kilodalton, but in lower quantity, 10 to 100 microgram per liter. In order to check this assumption, we perform some measurements on different synthetic mixtures. And to introduce a nanoside element, we use the beta-lactoglobulin protein, which was provided by Camille Lupiac from the Dijon lab. This protein has a molecular weight of 18 kilodalton, but when pH is between 3 and 7, which is the case of wine, it forms globular dimers of 36 kilodalton that should undergo a rotational diffusion with a correlation time corresponding to the observed dispersion. The simple mixture composed of water and ethanol do not reproduce wine dispersion. But when beta-lactoglobulin is added, the observed correlation times agree with the one measured in the wine sample. However, the concentration would be one order of magnitude higher than the one observed in real wine. Moreover, to observe the two singularities, we would have an insufficient amount. To observe these two singularities at 5 and 2 nanometer, we should have in a sufficient amount two narrow size distributions around 2 and 5 nanometer, which is unlikely to occur in wine. So in conclusion, the concentration and the size distribution of macromolecules or particles in wine would likely be insufficient to explain the NMRD profile features. So we had to put that assumption on the side and turn toward other possible explanation. The second idea came from the literature of the early years of NMR. On the right, we have a reproduction of the NMRD profiles of paramagnetic aquarium, manganese, iron, gadolinium, copper. What is noticeable on these figures is that paramagnetic ions give a dispersion between 1 and 10 megahertz. And moreover, manganese gives a second dispersion between 0.1 and 0.01 and 0.1 as in wine. So I turn to Professor Régis Goujon from Dijon, who is a determinant actor of this project. He is a specialist of wine. And I asked him, well, do we have paramagnetic elements, and in particular manganese in wine? He answered, yes, you may, but the amount would be low. And as far as manganese is concerned, you may find only few papers mentioning briefly manganese. Manganese in wine is not very studied. So let's, before going any further, let's open a second bracket concerning the sources of metals in wine. Metals in wine may come from both natural and anthropogenic sources, and their concentration is an important parameter influencing wine consumption and conservation. Indeed, metal ions play an important role in oxide-reducing reaction, resulting in wine browning, turbidity, cloudiness, and astringency. The quality of the wine is highly dependent on its metal composition. So there are two key sources which contribute to the presence of various metals in wine. The first natural source is the soil, where the vine is cultivated. Secondly, we have sources of human activity such as fertilization, phytosanitary treatment, enological practices, machinery, piping, use of finding agents, additives, environmental contamination by cars or factory, and also fraudulent practices. Iron concentration depends mostly on the ground where the grapes are produced, whether it covers the berries as part of the dust or whether it is absorbed through the roots. Aside from contact with copper or bronze materials during the winemaking process, pesticides are the main source of wine contamination with copper. I have to mention that additionally, copper is often added to wine because it eliminates smelly characters associated with organic sulfur compounds that may form during fermentation and aging. But it is worth noting that during fermentation, copper is reduced, forming insoluble sulfide, which in practice, which are removed during the final stage of the vinification with yeast and lees. In practice, we can end up with a wine containing less copper than the initial grapes. The vineyard appears to be the primary source of manganese in wine. Soil bioavailability and local environment factors tend to be the principal driver of manganese concentration. Vineyard activity may be able to reduce, but not to eliminate manganese level. And typically, the process of vinification does not add significantly to the levels already present in the grapes. Zinc wine content increases when zinc containers are used during the processing and aging. And also, of course, when zinc containing pesticides are used. Analysis of chromium concentration for various vintage wines of the same vineyard and winery showed that the content of chromium increased significantly with the wine age. And this may be attributed to contamination during storage by stainless steel container, rather than from other sources. Well, the use of clay bentonite for wine fining increased aluminum concentration in wine, but insecticides are also sources of aluminum. The use of brass, some type of brass component in winemaking, such as pumps, valves, faucets, and pipes, is the major contributors to lead contamination. But fortunately, the modernization of wineries using brass with stainless steel, hence reducing lead level in wine. Finally, the application of phosphate fertilizer is supposed to be the principal cause for cadmium contaminations of soils and wine. So I close the bracket, and what we did, we decided to analyze the metal concentration in two distinct French wines, a red Medoc from the Bordeaux and a white Chardonnay from the University of Burgundy. These wines differ from their vineyard, their vine, the technique, and the material used during the vinification. The only common point, perhaps, concerns the legislation relating to the use of phytosanitary products. So this slide shows the concentration of various metals measured by inductively coupled plasma atomic emission spectroscopy In purple, the red wine from Bordeaux, and in yellow, the white Chardonnay wine from the University of Burgundy. At first glance, and maybe surprisingly, the patterns are very similar. But we have to mention that the vertical scale is logarithmic. And it seems that we have always slightly more iron in the red wine than in the white one. The marked differences concern cadmium, but the level is so low that it's difficult to say a lot about it. Then copper, we nearly have a factor 10. Nickel, we have more than one order of magnitude, and lead, which also show a high discrepancy. If we are looking for an explanation, we may simply thinking in attributing this variation to the different material used during the vinification. For our study, the metal of interest are paramagnetic metals, particularly copper, iron, manganese, nickel. And firstly, to check if the wine profile originates from the paramagnetic metals in wine, we try to remove the paramagnetic ions by an exchange of the wine on a resin. This slide shows Chardonnay wine, the ion concentration, so on the wine before exchange in yellow, and in gray on the wine, the same wine, after exchange on the resin. The amount of metal decreases in the wine, but in different ratio according on the metal considered. The decrease in manganese is drastic from 1 milligram down to a few micrograms per liter. For nickel, it is also impressive, but the exchange is less efficient for iron, copper, and chromium. This may suggest that manganese or nickel have more affinity for the resin than copper or iron, but it may also suggest that in wine, manganese and nickel have less proclivity to form stable complexes that would limit the exchange with the resin. In the case of copper, for example, this is to put in perspective with the formation of insoluble copper sulfide previously mentioned. Okay, so on this slide, we report on the same scale the NMRD profile of the white wine before exchange and below after exchange. The profile has drastically changed. We removed nearly all the manganese and a lot of other paramagnetic ions. Dispersions are no longer observable. This is a nice correlation with the previous chemical analysis, and to further confirm that the paramagnetic nuclei are the main sink for relaxation, we try to simulate the profiles. Paramagnetic relaxation describes the interaction between the unpaired electrons of a paramagnetic nucleus with the surrounding protons. In solution, the paramagnetic nucleus here, manganese, is moving due to the Brownian motion, but also protons, which we may consider mainly associated with water molecules. One water molecule can come nearby the manganese, penetrate the hydration sphere of manganese, stay for a while, then leave, and so on. So doing, it interacts with unpaired manganese electrons. One of the primary and simple description of the interaction was proposed by Salomon. The system is modelized by a representation of the unpaired electrons at the center of the paramagnetic nuclei. and deals with the through-space interaction between a proton and the unpaired electron, like in the case of the BPP model. The calculation gives this equation, where the relaxation rate is expressed in terms of Larmor frequency of proton and electron and depends on some correlation times. Here, two contributions are reported. The first one is responsible for a dispersion in the range 1 to 10 megahertz. And the second one, which is not observable on the figures, appear at higher frequency. The dipolar constant is composed of parameters from proton and electron, and it contains the internuclear information. Correlation times have subscribed high because it depends on transversal and longitudinal directions. And it is a composition between the correlation time corresponding to the rotation of the manganese, the time the water molecule can stay close to manganese in the hydration sphere, by example, and correlation times from electronic relaxation. This model is able to reproduce the dispersion over the 110 megahertz, but do not reproduce dispersion at lower frequency. This means that it has to be completed with a second paramagnetic interaction that occurs when the electronic shielding is very delocalized, such as here in the case of manganese. This second interaction, described by the Bloembergen mechanism, takes into account the fact that the electronic orbital, here in green and purple, can physically extend up to the proton nuclei. This is a contact interaction. The expression of the relaxation rate resulting from such a contact interaction can be formulated as follows. The profile is described by a Lorentzian curve that reproduces well the dispersion at low field. So after taking into account a third contribution, a diamagnetic contribution that would allow to add some non-paramagnetic relaxation of the solution, the wine profile can be reasonably well reproduced. However, the wine contains other paramagnetic nuclei that are likely to produce dispersion in the range 1 to 10 megahertz. So we are now going to look at the impact on the wine profile of the other paramagnetic nuclei. This slide shows the relaxation rate versus the concentration of manganese, iron, copper, nickel for different concentration in model wine. If one considers comparable amount of iron, it is clear that only manganese and iron in oxidation state 3, iron 3, can compete for the relaxation. On the left, we reported the profile corresponding to the level of iron in the wine studied. It's about 1 milligram per liter. So for ferric and ferrous ion in green, ferric in red, ferrous in green, we see that ferrous ion would not introduce a significant dispersion, but this is all the contrary for ferric ion in red. These are the two extreme cases. The prime question is, what is the proportion of each iron ion in real wine? Well, it is well known that in a closed bottle, metals are in their low oxidation states, but if oxidation occurs, ferrous ions turn to ferric. It's a complex kinetic reaction depending on numerous factors, and it is not obvious to quantify the amount of ferric ion in the wine. But it is sensible to consider that a proportion of iron can be in oxidation state 3. It is probably low, but can contribute to some extent to the dispersion of the NMR profile of wine. This could explain, if necessary, the small discrepancy observed between the calculated curve and the experimental data. So, up to now, we removed the manganese ion and observed the disappearance of the singularity on the profile. Then we were able to simulate the data with the paramagnetic relaxation theories. In a third step, we made synthetic wine with various amount of manganese, and we obtained this blue profile for a wine containing 50 micrograms of manganese per liter and 1.28 milligram per liter of manganese. The right figure is an extension of the low content, the manganese low content wine. The dispersion observed in wine, well, the dispersion are very well simulated. And we can see that 50 microgram per liter is clearly above the limit of detection and quantification. And I have to add that these data were recorded with only 4 scans. So here are the numerical values of the refinements for the paramagnetic dipolar and contact interactions. For model wines with 5.5 milligram of manganese, 5.01, 1.28, 0.53 and 0.05 milligram per liter of manganese. The dipolar and contact constant vary nicely linearly with the concentrations, and the correlation times measured are very stable. I mean, it goes from 3.8 up to 3.4, except for the last sample. This may be due simply for the higher dispersion of the data. For each real wine, W for white, R for red, we did 2 measurements by 1 year of time delay. And for this measurement, we obtain also very realistic values. The correlation times are very close to the one observed in in the literature concerning manganese in water. So based on the previous observation, we are now going to try to quantify manganese concentration in wine. The idea is to take advantage of the low frequency singularity that originates purely from manganese relaxation through the contact interaction, and so to be free from other contribution coming from other paramagnetic ions or from different diamagnetic relaxations that may vertically shift the wool profile. So on the left, we reported the titration curve of model wines containing different amount of manganese for the different Larmor frequency used for recording the natural wine profile. For each frequency, relaxation rate is linearly dependent on the manganese concentration. The slope depends on the Larmor frequency considered. And providing that we are not working at too high frequencies, the intercept, which correspond to the relaxation rate when there is no manganese, can be considered as the same in all cases. So if we choose a point A at a given frequency, we can write the former relation. And if we take a second point B at another frequency, we can write the equivalent relation. So we can then solve these two equations to extract the manganese concentration, which can be expressed as the difference of the relaxation rate of the 2 points divided by the slopes of the titration curves that depends on the Larmor frequencies. If the 2 points are chosen where mainly manganese drives the relaxation mechanism, one can expect to be able to extract the manganese concentration. So we did this calculation for all possible couple of points, and the results are reported on the contour plot on the left. It shows the calculated manganese concentration for all possible couple of points of a model wine containing 0.53 milligram manganese per liter. The contour plot is symmetrical with respect to the diagonal, so only half of it is reported. To be more precise, let's consider all the points of the domain A and all the points corresponding to B. So this, if we have, let's say 4 and 5 points here, reported on the contour plot, will be calculating 4 by 5, 20 concentration. And we obtain an average value of 0.53 and a standard deviation of 1. 1 is on the last digit. Okay. So we did the same calculation with a larger domain for B, which corresponds to the rectangle here, and we obtained the same way an average of 0.52 with a small standard deviation. We even did the calculation on a larger scale in this triangle, which is difficult to reproduce here. And the calculated value remains very close to the true value, but with a higher standard deviation. Okay, so we tried the same calculation on the real wine. And I will now show the contour plot of the real wine. I mean, the figure is quite different. This is the white Chardonnay wine that contains 1.26 milligram per liter iron measured by ICP. Well, the contour plot, on the contour plot, the variations is more pronounced. And the estimation with the same surface as previously, I mean, these two narrow domains that correspond to the square give values of 1.17, quite close. And if we extend we stay with a very reasonable value, 1.2, and even in the triangular area, we stay very close with 1.3. Okay, so here are all the results for 5 model wines with the concentration, synthetic concentration, and the measured concentration by relaxometry. So in all cases, we are very, very close. But in fact, these model wines only contain manganese. There is nothing else but manganese. So we are lucky that it works, but fortunately, I would say. For the real wine, so as I said previously, white wine and red wine measured with a difference of 1 year. So we have seen the result for the white wine. There is no lot of change. The measurement on the red white seems to underestimate a bit the manganese concentration. But well, we may find many explanations, I think, for that. We also did the same calculation on the exchange wine, where we have very little remaining manganese. ICP give a maximum of 2 microliter, 2 microgram per liter of wine of manganese, and we measured, I mean, something between 9 and 10 microgram of manganese per liter. So still, we have a reasonable value, even for on the exchange wine. So if you want more wine, well, this is not the nicest slide, but it's just a compilation of different profile, different vintage, different type of wine. We even have a champagne in this. And well, in all cases, so far, we have always observed these 2 singularities due to manganese. I mean, every time it appears. I mean, this one is of poor quality, but I mean, the tendency seems to be there. So, in conclusion, manganese in wine comes primarily from the soil. Its concentration is not affected by wineries' practices. It does not seem to form many stable complexes. Manganese seems to be observable on many types of wine. So, manganese could be a valuable fingerprint for wine. And relaxometry that could allow an easy and precise in situ concentration measurement would then be a technique of choice for wine control or identification. So most of the slides were taken from a paper we published with Adam Lashoki and Jadwiga Trickott from Poznan, Bernard Michael and Philippe Schmidt-Koplin, who did the ICP measurement in the Analytical Biogeochemical Center in Munich. Régis Goujon and Thomas Carbeviac from Dijon. Thomas Carbeviac is the leader of the team, and he was a great support and made also all this project feasible. And then I want to thank also some master students who worked more or less on this topic, Emeric Batlog and Manuela Alakik, and PhD student, Pierre Fouillou and Susanna Esoyan. And I want to say that prior PhD student, Susanna was working in the Areni cave in Armenia. And she is now doing her PhD in coordination with Nelly Owanisyan, who wrote a beautiful book on Armenian wine and vine, and from where I took the first slides. So I think that's all I have to say for the moment. So Thank you for your attention and cheers.